Hi, dear friends and cultural creatives. Bruce here again. This time I want to talk about the topic of epigenetics. Well, most of us are familiar with how epigenetics controls gene activity and also modulates gene expression. Many people are really unsure as how does this process actually work? And this becomes very important because otherwise we're just talking about a new age belief. Yes, epigenetics changes your heredity expression and all that. And yet I really want to make it clear to you how it works because knowledge is power. And once we have knowledge of this, we'll have more power in regulating our own genetics and our epigenetics. So let's start with the first premise. Understanding epigenetics. Epigenetics, as the name implies, epi means above, genetics is the genetic activity, so epigenetics means actions above the genes. This is where the source of the control of the genes come from. And this is very, very important for after 50 years of describing life that genes make decisions and genes turn on and off. We're really beginning to come to a very clear understanding that genes are just simply blueprints. And as a blueprint, they have no on and off. And why this becomes important is rather than directing our attention to the DNA for activity of our genes, we really have to look elsewhere. And what we're really looking for is where's the contractor of the cell that controls the genes. Well, those of you familiar with the biology belief have recognized that we move the brain of the cell from the conventional understanding being the nucleus actually to the cell membrane, the skin of the cell. And why this is important is because the skin of the cell is the interface between the environment on the outside and the environment on the inside. So when I get environmental signals coming from the world around me, the membrane picks up these signals and then transfers information about the environmental signals into the cell. As the information leaves the membrane and goes into the cytoplasm, the information can either influence the proteins in the cytoplasm, which create the biological functions such as respiration, digestion, and excretion, or the environmentally relayed signals can go into the nucleus where they affect the hereditary material. So to understand how the environmental signals impact and alter genetic activity requires us first to understand the nature of what a chromosome is. A chromosome is a hereditary unit. It's 50% DNA and 50% protein. Well, for 50 years, we've been so focused on the nature of the DNA that we've really ignored and just actually discounted the role of the proteins in the chromosome. So now what I would like to talk about is the nature of the DNA and the chromosomal proteins built into our genome. As we now know, there are approximately 21 or 22,000 genes in the human genome. This accounts for less than 3% of the total amount of DNA. For the longest time, scientists held that the remaining 97% of the DNA was called junk DNA because it didn't have a role in creating proteins, so it wasn't being used as a template for protein synthesis. People thought it was junk because they thought it was just DNA that was continuously carried forth through evolution, not having any particular function, so it was just being accumulated in the nuclei of our cells. I always get a slight bit of humor out of the situation because humans are basically very inefficient, and then when we look at cells, we apply the nature of our inefficiency to cells, saying, yes, the cells are just accidentally carrying around millions of years of DNA that it's not using, and this is called the junk DNA. Well, nothing can be farther from the truth. The junk DNA, in fact, is the most important DNA in the genome for a simple reason. It's the junk DNA that is where the regulation of the genes comes from. And what we are now beginning to recognize is that on this junk DNA, there are protein modules. So, for example, in the illustration that I'm going to show you right now, this is a segment of DNA. A portion of the segment, as illustrated in the boxed-in area, is the actual part of the DNA that represents the gene program, the blueprint to make a protein. The DNA in the front of that gene, or in the DNA following the gene, is conventionally listed as junk DNA. But now we recognize it's the proteins on this junk DNA that form little groups or clusters of proteins called modules. These are the modules, when activated by environmental signals, that control the fate of the gene blueprint. To give you an understanding of this and to also emphasize one of the most important aspects of epigenetics, and that is this. Epigenetic mechanisms not only activate or inhibit genes, but they can alter the expression of the gene blueprint. But the most important understanding is this. Epigenetic alterations of the gene blueprint don't change the original gene. They modify the readout of the gene, but without changing our own genome. So that while we were born with a set of genes and die with a set of genes, the expression of those genes is a variable based on the environment, and this variable does not change the genetic code at all. So if you were born with a mutant gene, you'll always have a mutant gene, but you may not express it under appropriate epigenetic signaling. You say, how can this occur? Well, to give you a model, let's go back to the old days when television wasn't on 24 hours a day. And when we look at the screen of the television after the broadcast day is over, generally there's a pattern called a test pattern as illustrated in this picture. Well, what's significant is the test pattern is a pattern. The pattern is like a gene. A gene is a pattern. And now what I'm trying to get to is this. The pattern is broadcast to the television set, right? So that's the incoming pattern. But the dials on the television set that include on and off, volume control, contrast, focus, hue, color intensity, all of these dials can be used to modify the image of the test pattern on the screen. So as I show in this image, we can modify the horizontal and vertical hold and distort the picture. And most important question is this. Yes, the picture on the screen is distorted by changing the dials on the television set. But the most important question is this. Did I distort the original broadcast pattern? And the answer is absolutely not. The original pattern is there, but via the dials of adjustment, I can change the appearance of that pattern without changing the original broadcast. Again, look, I can change some other dials and again distort the picture of that test pattern. And yet, same thing. I've altered the expression of the pattern, but I didn't change the pattern. So there's a parallel analogy between epigenetics and the television set and the dials on it. So what this really means is the junk DNA part has switches and modules of protein that are the equivalent of the dials on the television set. When signals from the environment bind to these protein modules in the junk DNA region of the genome, they modify the readout of the proteins, which in turn adjust and modify the readout of the genes. So basically, by altering the protein modules in this junk DNA, I can alter the expression of genetic activity. Just recently, science has found there are 400 million switches that control gene activity that are in the junk DNA. There are 400 million dials, so to speak, that can adjust the readout of our gene activity, creating a tremendous variation, creating thousands of different versions of a blueprint from a single gene original blueprint. So now what we're looking at is the DNA. Part of it is the blueprint for the gene. The massive part of the DNA, called the junk DNA, is the regulatory agent of our genes. And this is why it's so important. If you alter the regulatory portion, you alter the readout of the genes. So you may be born with a healthy set of genes, but if you negatively influence the genetic readout via the junk DNA, then that's through environmental signaling. You can take a normal reading gene, modify its expression so that it reads as an abnormal gene. Now, of course, the big question is this. How can the signals from the environment alter the genetic activity without changing the blueprint of the gene? Well, as we're going to illustrate in the next section, a gene is like a ticker tape, an old-fashioned ticker tape where information is put out letter by letter by letter along a single strip. So as the ticker tape device rolled out a piece of paper strip, you could read the words along the ticker tape. Well, consider a gene a linear piece of ticker tape, and it's got words on it which represent the gene blueprint. So in the model that I will show you, consider that we copy the gene, and that's called the messenger RNA. And let's say we'll use a gene called the reading gene. So our little ticker tape of DNA has READING as the genetic code on it. And I say, well, what can I do with that? Well, when I make a copy, now I have the messenger RNA, it's called, which is the copy of the original gene with the READING written on it just like it has in the original gene. And then I say, well, what does epigenetics do? And here's the interesting and exciting part about epigenetics. Epigenetics is like taking a pair of scissors and tape and cutting up the messenger RNA into segments. And as you cut it up into segments, you can retape the segments in different sequences and change the coding of the DNA in the messenger RNA. So as we can see in the following illustration, we can take the word reading and split it and turn it into the word read, get rid of the ing. Or we can take the word reading, get rid of the REA and leave the word ding. Or we can keep cutting and modifying the signal as read through this linear strip of messenger RNA, cut it and reassemble it and create a bunch of different words. So just in the model that we're illustrating on the screen right here, what we can see, there's 5 or 6 different versions of the reading gene, all resulting from cutting and taping together the pieces and creating new words. Well, this model is actually what goes on in our genome. We copy the messenger RNA version, and then the information from the epigenetic modules, the proteins in the junk DNA, determine where the gene should be cut and how it should be reassembled. And so if you can just look at the simple reading gene that we've illustrated right here, and you can see how we can make so many variations, then you can get an insight into a DNA strand which is thousands of letters long and being cut up and reassembled in different sequences, giving me a blueprint, the messenger RNA version of the blueprint, with many different organizations of the code, providing for different developments of proteins without changing the original DNA. So basically, epigenetics is a mechanism where environmental signals engage especially the regulators in the junk region of the DNA to adjust and modify the readout from our genes. And therefore, we can understand how a single gene blueprint can be used to create thousands of different versions of proteins just by cutting and reassembling the gene. This is the most important understanding about epigenetics because it simply says this: irregardless of the genes you were born with, you can change the readout of those genes via epigenetic mechanisms which are controlled by environmental signals and our perception of environmental signals. Which means, while you received a set of genes, you are not necessarily a victim of those particular genetic programs, because by the way you respond to the programs, you can rewrite those genetic programs. Now, unfortunately for most of us, this is a problem for a very simple reason. 95% of us were born with gene programs that would provide for a healthy, happy, prosperous life on this planet. Normal genes, 95% of us. But the issue is we start to recognize that through our interaction with the environment, we can modify the readout of those genes, and the modifications almost generally lead to a less than happy solution to the reading of the genes. We can alter the readout of the genes and create mutated proteins. The mutated proteins lead to issues such as cancer. So we're beginning to realize, well, cancer has been always thought to be due to cancer genes. The new insights have recognized that cancer is primarily due to altering the epigenetic protein modules and then altering a normal gene and then resulting in a pathology. So as we've been blaming the genes for our illnesses and the problems that we face in our lives for the last 50 years, it turns out that we've given them a bad rap. That the bigger issue we have to deal with is recognizing we are the ones that are rewriting our genes. And we rewrite our genes in harmony or in disharmony with our perceptions of life. And so basically, the whole new understanding of epigenetics says this. Old version of genetics said we were victims of the gene programs we received from our parents. The new story of epigenetics is a complete turning of the table on that. It says that we were given a set of genes and we have the power to rewrite the genetic expression of those genes. And all of a sudden it says we're not victims of the genes, we're masters of the genetic readout. But mastery also implies something that we must wake up to, and that is called responsibility. Because it is we who control our gene activity. When we have negative thoughts, when we have disempowering beliefs, when we have self-sabotaging behaviors, it's these activities that are directly interacting with our genetic readout via epigenetics that determines our biological fate and actually determines the health and pathologies that we face in this world. So this is why it is so important for the public to start to understand the nature of epigenetics for a very simple reason. It's the nature of how we control our own biology, how we are not limited or prevented from expressing the best quality of life on this planet, other than through the use of our own mind and our own perceptions. And this is why I'm so happy to give you this mechanistic information, because I don't want it to look just like New Age, epigenetics controls our biology. I want to show you how it does that. Knowledge is power, and I hope this knowledge has been empowering to you.